Hi friends, today's our topic structure of microsporangium and microsporogenesis. First I wish to briefly explain the pre-fertilization structures and events. Several hormonal and structural changes result in differentiation and development of the floral primordium. All flowering plants are the angiosperm, show sexual reproduction. Flowers are the site of sexual reproduction. Androecium is the male reproductive part of the flower. It consists of the wall of the stamens. Gynoecium is the female reproductive part of the flowers. Androecium consists of wall of the stamens. The number and length are variable in different species. The parts of the stamen, it is divided into two parts, filament and anther. Filament is long and slender stalk. It's proximal and is attached to the thalamus or the petal of flowers. Anther, terminal and typically bilobed. Each lobe has two thecae, so it is called dithecous. A longitudinal groove runs lengthwise separating the theca. The anther is a four-sided, so it is called tetragonal structure, consisting of four microsporangia located at the corners, two in each lobe. The microsporangia develop further and become pollen sacs. They extended longitudinally and through the length of an anther and are packed with pollen grains. Next we discuss about the structure of microsporangium. Each microsporangium surrounded by four layers: epidermis, endothecium, middle layer and tapetum. The outer three layers gives protection and helps in dehiscence of anther to release the pollen. The innermost layer is tapetum, which is multinucleated with dense cytoplasm. It nourishes the developing pollen grain. The center of each microsporangium contains homogeneous cells called sporogenous tissue. The process of formation of microspores from pollen mother cells through meiosis is called microsporogenesis. The sporogenesis tissue of the microsporangium differentiated into microspore mother cells or the pollen mother cells. Each microspore mother cells undergo meiosis and give rise to haploid microspore tetrad. Microspores are arranged in a cluster of four cells, so it is called microspore tetrad. On dehydration, microspore tetrad dissociate into form four microspores. Each microspore developed into a pollen grain. Next we study about the pollen grain. Pollen grain represents the male gametophyte. It is spherical and measuring about 25 to 50 micrometer in diameter. It is covered by two layers. The outer layer is the exine and the inner layer is the inteine. First we say about the exine. The hard outer layer called the exine is made up of sporopollen, which is one of the most resistant organic material. It can withstand high temperature and strong acid and alkali. Enzymes not degrade sporopollen. The exine has the prominent apertures called the germ pore, where sporopollen is absent. Thus, the inner wall is the inteine. It is the thin and a continuous layer. It is made up of cellulose and pectin. Wall of pollen grain, that is the inteine, consists of two cells, the vegetative cell and generative cell. Vegetative cells, it is a bigger, abundant food reserve and large irregularly shaped nucleus. Generative cells, small and float in the cytoplasm of vegetative cell. Spindle shaped with dense cytoplasm and a nucleus. These are the characters of vegetative cell and generative cell. 60% of angiosperms, pollen grains, are shed at this two-cell stage. In other, the generative cells divides mitotically to form the two male gametes before pollen grains are shed at the three-celled stage. These are the characters of the pollen grain. First we explain the structure of microsporangium, then microsporogenesis, then pollen grains. Okay, thank you.